Today's presentation is using clinical pharmacology and pharmacometrics to accelerate cell therapy development, a patient's CAR-T journey. Next slide, please. Our speakers are Dr. Catherine Brown and Miriam Traim. So let's tell you a little bit about our esteemed speakers. Catherine is currently a senior director in the Integrated Drug Development Group at Certara, where she leads the complex biologics practice area. She has more than 20 years of experience in clinical pharmacology and pharmacometrics in the pharmaceutical industry, and she earned her doctorate in physical organic chemistry from the University of Durham in the United Kingdom. Joining Catherine is Miriam, who's a senior director in the IDD group at Certara, where she is the Northeast regional lead for our Quantitative Services Solutions group. Miriam has more than 12 years of industry and academic experience in clinical pharmacology, pharmacometrics, systems pharmacology, and more than 3 years of experience in cell and gene therapy. Miriam earned her PhD, her PharmD from the University of Florida, and her PhD in clinical pharmacology and pharmacometrics from the University of Münster in Germany. Catherine and Miriam, welcome to today's event. I'll now turn it over to Catherine to begin the presentation.
Great, thanks Suzanne, and welcome everybody. So if we can go to the next slide, please. So today we're going to talk about why there is interest in developing cell therapies. We're going to take you through a typical patient's CAR-T journey, and then we're going to talk about some of the upcoming developments that are happening in this ever-evolving field. So next slide, let's start with why there's an interest in cell therapies. So on the next slide, we're going to specifically focus on CAR-T cell therapies in oncology today, as there's been a number of approved therapies in this space in recent years. And these have really shown some distinct advantages over other types of drugs used in oncology. They have really been transformative for patients who have cancers that are very aggressive and where other standards of care have failed. Cell therapies also carry with them the prospect of longer duration of response, which means that patients remain stable for much longer. And these transformations in care are really down to how cell therapies work. So they are immunotherapies, which basically refers to agents that prevent or treat a disease through affecting the immune response. And T cells are a type of lymphocyte that are an important part of the immune system. So shown on the left here is a schematic of how a CAR-T cell therapy works. So we start with T cells being removed from, in this case, the patient themselves, followed by genetic modification to generate CAR-T cells. So CAR stands for chimeric antigen receptor, and it basically means that a gene is added in, and it's a gene that enables recognition of a specific antigen known to be expressed on the tumor cell surface. So these CAR-T cells are then expanded to give sufficient numbers and then reinfused back into the patient. So once in the patient, the CAR-T cells then seek out and bind to the antigen expressing tumor cell, as shown in the right-hand diagram here. And this then promotes cell death by initiating signaling cascades. It promotes T cell activation, proliferation, and production of cytokines and chemokines. So the CAR-T cells can persist in the body for a long time, so that's why there's the potential to recognize and attack cancer cells if the patient relapses, which then provides that longer duration of response. The administration of a cell therapy is typically a single infusion, or in some of the newer cell therapies being developed, it's perhaps a couple of infusions within the first month, and then essentially that's it. So it's really a one-off treatment, which is definitely also preferable from a patient's perspective. So on the next slide, just some of the history of the development of cell therapies. So starting way back in 1960, when it was recognized that immune cells can kill cancer in mice. At that time, it wasn't completely understood in terms of the exact types of cell involved, but that understanding developed over the next 20 years or so, when T cells and their importance became more understood. So in 1973, T cells from a bone marrow donor killed cancer cells in the recipient, and that was one of the first successful immunotherapies. And then in 1986, it was shown that a patient's own immune cells can fight their cancer. And all of this knowledge came together then in 1992, when T cell engineering began, specifically using retroviral vectors, which is where you use RNA from a virus as a way of introducing specific genes into the T cells. That then led to the first generation CAR-Ts. Those didn't persist in the body though, and so further development resulted in the second generation CAR-Ts, and then subsequently later generation CAR-Ts, and those include a co-stimulatory molecule. So in this case, we have CD28, and that essentially augments the ability of the CAR-T to proliferate and persist in the body. The first CAR-T targeted against the CD19 antigen was then developed in 2003. The first patient dosed in 2010, and such positive efficacy was seen that the FDA granted breakthrough therapy designation in 2014, and the first CAR-T therapies were then approved by the FDA in 2017. On the next slide, you can see that there are now 6 therapies approved, and they're listed here on the left in the blue box. If we move to the orange box, you can see that 4 of the approved therapies target the CD19 antigen, which is widely expressed on B-cell malignancies and not really expressed on other tissues, which makes it a good target for cell therapies. 2 more recent approved drugs target the B-cell maturation antigen, which is a common cell surface antigen in multiple myeloma. If you have a look at the indications in the green box, you can see that they're all hematological or blood cancers. And solid tumours are really harder to target when it comes to CAR-T cell therapies, as they need to be able to penetrate the tumour. But the antigens and the chemokines needed to promote that type of infiltration isn't really expressed sufficiently by the tumour. So these CAR-Ts are restricted to blood cancers. Some of the newer types of cell therapies are looking to find ways to effectively treat solid tumours, and we'll touch on some of those a little bit later. You can see in the red box there that all are approved as later line treatments. So they're used in hard to treat aggressive cancers, and they have been demonstrated to work where other therapies haven't. We do also need to bear in mind though that cell therapies do have some safety risks associated with them, and they're also expensive. So they wouldn't be the first port of call for a prescriber. In terms of dose, so shown in there in the purple box, some are dosed as a flat dose, others are dosed mg per kg, so by body weight. And coming up, we'll discuss a little bit later how modelling can be used to assess which is the most appropriate for a particular therapy. And finally, on this slide, all of the approved CAR-T shown in the blue box here are autologous, which means that the T cells that are engineered are T cells taken from the patient themselves. The landscape is starting to change though, and it could be that some of you in the audience today are working on allogeneic therapies where the T cells are taken from a healthy donor rather than the patient themselves. So let's see if that's the case or not. We're going to go to a poll and we're going to ask you a question. So if you can answer this, so do you work on autologous or allogeneic cell therapies? So please select one of the options: only autologous, only allogeneic, both, or if you don't work on either, please indicate that. Okay, great. So majority in the audience are pretty new to cell therapies, but I can see that's interesting. A lot of you are working on actually both, and quite a few, 5% allogeneic, 13% on both. So although all of the approved ones today are autologous, there is obviously some activity there in terms of allogeneics. So let's have a look on the next slide. Let's compare autologous and allogeneic cell therapies. So as mentioned, autologous means the patient donates their own T cells. Allogeneic means a healthy donor donates the T cells, which means that one donor can then help multiple patients. And the advantages of autologous therapies is that you get prolonged persistence, and typically standard lymphodepletion is sufficient. Now, Miriam will talk a little bit later about lymphodepletion, but just to say at a very high level, it's a treatment that the patient receives prior to the cell therapy to really prepare their immune system for receiving an influx of the T cells to help promote the ability of the CAR-T cells to expand and then replicate after injection. So those are the advantages, but there are some disadvantages of autologous therapies. There's a high risk of CRS and ICANS, which are the two main AEs related to the use of CAR-Ts. So CRS is release of cytokines that are activated by the expanding T cells, and ICANS is a neurotoxicity. It's also a more complex and certainly lengthy process for the patient because they have to wait while the CAR-T cells are manufactured, as opposed to an allogeneic therapy, which is essentially off the shelf. There's also more variability, so not all patients are eligible as well, and it's a more expensive process. So allogeneic therapies are attractive because they address the majority of the disadvantages associated with autologous therapies. However, they do come with some disadvantages of their own, namely that they typically require higher doses of those lymphodepletion therapies to prepare the patient for receiving the cells. And they also carry the risk of graft versus host disease, where the patient can potentially reject the donor cells if they aren't sufficiently matched. So if we go to the next slide, we've talked a little bit about the changing landscape in terms of autologous versus allogeneic. And the regulatory landscape is also evolving. So if we start with the FDA, so cell therapies are regulated as a gene therapy within the Office of Tissues and Advanced Therapies, or the OTAT, and it's in the Center for Biologics Evaluation and Research, so CBER. The agencies are also adapting and helping and working with sponsors to really understand these relatively new types of therapies. For example, OTAT is running a series of town hall Q&A sessions to cover various topics. The first one's going to be at the end of September, and they're going to cover CMC aspects. The FDA have a number of guidances on their homepage, shown here on the left, including their most recent guidance from March 2022, which is shown here on the right. And that's the first guidance to specifically talk about CAR-T cell therapies, and it covers manufacturing aspects. It talks about characterization of cellular kinetics and pharmacodynamics. It also covers dose selection, and specifically, interestingly enough, the recommendation to dose by weight or body surface area, rather than using a flat dose, based on the FDA's experience of how safety and efficacy can be influenced by body weight. If you recall a few slides ago, some of the approved CAR-Ts are given as flat doses and others by body weight. And we'll come back to that later and how modeling can be used to assess the relationship between body weight and outcomes. So on the next slide, just moving to EMA. So within EMA, the cell therapies fall under the Human Cell-Based Medicinal Products Guidance shown here, which came into effect in 2008. And whilst it's not specific to CAR-Ts, it does give some insight into the types of information that EMA will be looking for at the point of submission. So on the next slide, I'm going to move on now to talk a little bit about the commercial aspects related to CAR-Ts. And really it's a balance, because there are great opportunities and patient benefits for transformational treatments. On the other hand, it is a process that the patient needs to go through, which particularly for autologous treatments is quite a burden for the patient. And there is a time and cost component associated with these therapies. So what we've found here is that really thinking about these complexities sooner rather than later, so really even before you go into the first clinical trial, that's advantageous in smoothing that development path and ultimately the patient experience. So let's go to the next slide and have a look at what that patient experience is like by following a patient's CAR-T journey. So we're going to follow both an autologous and an allogeneic path so that you can see the differences and the similarities. There are some common aspects, as you can see, where the two paths converge over on the right. The lead up to the lymphodepletion step differs though. And you can see straight away from the schematic the length of time from identifying an eligible patient to starting that lymphodepletion step is significantly longer for the autologous treatment than for the allogeneic treatment. So patient eligibility is at the far left box at the top, whereas at the bottom it's the step just prior to lymphodepletion. And that difference in time is not unsubstantial when we consider that these are pretty sick patients waiting to be treated. Let's take each of these steps in turn though, starting with the patient eligibility. So let's move on a couple of slides. So for autologous therapies to be eligible, the patient needs to have sufficient viable T cells to enable collection, and the number of viable cells can be affected by the disease or prior treatment that the patient has received. For allogeneic therapies, there typically is a screening process to enable matching between the donor and the patient to prevent the graft versus host disease where the patient can reject the donor cells. And donor matching can involve what is called HLA matching, where HLAs are human leukocyte antigens that are found on most cell surfaces, and they're important in modulating immune responses. So HLA matching between the donor and patient can minimize the risk that one set of cells tries to attack the other. Listed below then are the eligibility requirements that are common to both autologous and allogeneic therapies. So mentioned previously, CAR-Ts are associated with hematological cancers, and they target agents on the cell surface, and some of those examples are shown here. Generally, the patient also needs to be well enough to receive the cell therapy, and as mentioned before, these patients will have received multiple other therapies previously. So next on the journey, on the next slide, is leukapheresis, which is then the actual collection of the cells in either the patient or in the healthy donor. And on the next slide, you can see essentially the process is the same for autologous and allogeneic. If we can just go back one slide. And there are some pros and cons to each. The main pros for autologous therapies is that using the patient's own cells removes the risk of GVHD, which we just talked about. And generally, as long as the patient has sufficient viable cells, then the process can be completed. One of the main cons, though, is the fact that the patient has to actually undergo this procedure and then wait whilst the CAR-T cells are manufactured, which does take some time to complete. And that's really one of the main pros of the allogeneic treatment, as shown on the right, that the cell product is readily available, and because they come from healthy donors, they're really robust T cells. The main cons we've touched on already around the potential need for matching and the chance of GVHD. So after leukapheresis, on the next slide, you can see that for autologous cell therapies, the patient then normally receives what we call bridging therapy, and that's to help control their active disease while they wait for the CAR-T cell manufacture. So this step is specific to autologous cell therapies. If we go to the next slide, just to learn a little bit about bridging therapies. So as you can see mentioned in the third bullet here, bridging therapy can be different between patients, which can then introduce variability. So when we're assessing the efficacy of the cell therapy, it can confound the observations. So we typically assess the impact of bridging therapies when we look at efficacy. And there are also a few considerations there listed in terms of ensuring the bridging therapy is appropriate for a patient who's just about to receive CAR-T therapy. So on the next slide, we're going to move on to CAR-T cell manufacture, and I'm going to hand over to Miriam to take you through this and the rest of the patient journey.
Thank you, Catherine. So the CAR-T cell manufacturing, as we see on the next slide, starts by collecting the patient's or the donor stem cells via leukapheresis, during which the PBMCs are isolated. The leukapheresis is then followed by a selection process during which the T cells are isolated and activated after genetically transduced with a CAR encoding transgene in vitro using lentiviral or retroviral vectors. Following the transduction, the T cells are expanded in vitro in the presence of the specific cytokines to replicate and multiply, and then the formulation and the cryopreservation of CAR-T cells is followed by infusion after product release testing. And there are two distinct differences during the transduction phase between the autologous and the allogeneic CAR-T cell therapies. So for the autologous CAR-T cell therapies, the CAR gene is actually transferred into the T cells, whereas for the allogeneic CAR-T cells, we're looking into genome editing of the T cells, and then the CAR gene is transferred into the edited T cells. And on the next slide, we're going to discuss a few key aspects which we need to keep in mind, which are important for dose selection. So one key aspect is the cell fitness and the donor variability in the cell material of the autologous CAR-T cells, what Catherine already alluded to a little bit. The number of the viable therapeutic cells is limited by the disease or the cytotoxic treatments the patients have received previously during the autologous CAR-T therapies. Patient age, immune system, and the disease state can also affect the expansion and the reproducibility between the cell therapy batches. And the transduction efficiency is another aspect we have to keep in mind, and this describes the percentage of the transduced cells. This transduction efficiency can actually vary between the manufacturing lots, which then results in differences in the active cell dose between patients for the same total cell dose. And hence, the dose should be based on the number of the transduced CAR-T cells in the drug product rather than the total cell number. Another aspect is our vector copy number, which is determined as a percentage of a CAR-positive cells, and the VCN as a function of the CAR-expressing cells will provide a more accurate representation of the VCN in the transduced cells. So after the manufacturing has completed, we see on the next slide, the patient now undergoes a lymphodepletion. But before we go there, we would like to introduce a poll question. So we would like to know, do you already dose optimize your lymphodepletion regimen? Please select one of the answers, yes or no. So this is an interesting answer. So many have answered no to dose optimization of the lymphodepletion regimen, but it's great to see that some are actually looking into this. And on the next slide, we're going to start talk about why the lymphodepletion optimization is key to CAR-T efficacy. So patients receive lymphodepleting chemotherapy a few days prior to CAR-T infusion to improve the immune environment, promoting the ability of the CAR-T cell dose to expand and replicate after injection. And common treatments for autologous CAR-Ts are cyclophosphamide and fludarabine. And for the allogeneic CAR-T therapies, a monoclonal antibody is actually added to the lymphodepletion regimen to overcome the potential alloimmune rejection by the NK cells. Lymphodepleting conditioning regimen likely works by multiple mechanisms, including eliminating the sinks for the homeostatic cytokines, which actually results in an increase in cytokines such as the interleukin-2 and interleukin-6, as well as interleukin-7 and 15, which have been already correlated positively to CAR-T cell expansion. It also eradicates the immune suppressive cells, such as the regulatory T cells and the myeloid-derived suppressor cells. It modulates tumor cells, as well as that it promotes the CAR-T cell expansion, function, and the persistence. And if not dose optimized, lymphodepletion results in lymphopenia, and it can also lead to neutropenia, thrombocytopenia, anemia, as well as immunosuppression. In addition, the choice of dose of lymphodepletion treatment can actually impact the performance of the cell therapy. And it has been shown that higher intensity of lymphodepletion regimen is correlated to a favorable cytokine profile, resulting in higher day zero MCP1 and higher peak interleukin-7, as well as a favorable outcome post-CAR-T treatment in terms of a higher response rate. And it has been shown that a lower intensity lymphodepletion, on the other hand, is actually resulting in an unfavorable cytokine profile, so lower day zero MCP1, lower peak interleukin-7, as well as an unfavorable outcome of CAR-T therapy. So how can we best dose optimize and model the impact of lymphodepletion regimen? Let's look at the next slide at a proposed study design for a phase 1 study. Because optimization of a lymphodepletion regimen is critical for CAR-T cell therapy efficacy and will be held to Project Optimus requirements for new chemical or repurposed entities by the FDA. So especially for the allogeneic CAR-T therapies where we include an antibody, the antibody together with the fludarabine and the cyclophosphamide will interact to form the overall lymphodepletion response. And this kind of interaction must be dose optimized systematically in the patient population while optimizing the CAR-T cell dose. So definitive screening designs, which fall under the umbrella of optimal design, are a class of design of experiments that can efficiently detect and quantify the effect of each component and interaction of each effect with minimal confounding. So if we look here to the left in this table, we can actually study 3 lymphodepletion components and the CAR-T dose at 3 different levels each, with only 9 cohorts in a trial. And at each factor level, all other factors are actually balanced and reduce confounding as shown in the white space on the confounding map. And as output, we can look at these correlation plots between the lymphodepletion response and the respective dose of each component in the study design and can hence determine the best dose regimen for our lymphodepletion agents in correlation with the cell dose. If we look at the next slide, after successful lymphodepletion, the patient is ready to receive the CAR-T drug product. Next slide, please. So after infusion of the drug product, we can see this kind of typical profile of a cellular kinetics. And unlike conventional drugs, CAR-T cells typically proliferate within the recipient, which results in a highly variable relationship between dose and exposure. So CAR-T cells are administered via IV infusion, which is followed by a redistribution of the cells in the body into different tissues, including the tumor sites, within a very few hours. And this is observed, as you can see here in the figure, in this initial drop of the circulating levels of the CAR-T cells. This initial redistribution phase is then followed by a phase of rapid expansion of the CAR-T cells due to the activation and the proliferation of the CAR-T cells. And the circulating CAR-T cells frequently peak within 2 weeks of administration, after which they decline at a variable rate. So to measure the CAR-T cell exposure, we use maximum CAR-T cell levels, so the Cmax, as well as the area under the concentration curve from day of infusion, so day 0 to day 28. And both the Cmax as well as the AUC reflect actually the early CAR-T cell expansion and are associated with clinical response to the therapy. In some recipients, CAR-T cells persist long-term, and this is observed for example, for the anti-CD19 CAR-T cell products by the persistent depletion of normal B cells, just the B cell aplasia which we are observing, which is an on-target and an off-tumor toxicity, which can persist longer than up to 4 years after therapy, although these persistent rates are variable and may depend on the CAR target, the co-stimulatory domain within the CAR-T cell phenotype, as well as the patient and the disease characteristics. So what is the optimal dose, and why is it so important to look for this? So on the next slide, we outline that the justification of the proposed to be marketed dose range is actually evaluated by the FDA as part of the clinical pharmacology and the pharmacometrics review in a very similar fashion as for small molecules or other biologics. So finding the optimal dose in CAR-T therapy is challenging but crucial. And hence exposure safety and response evaluation are central to dose justification. Let's talk about the safety first. Following a CAR-T cell infusion, we usually experience an onset of cytokine release syndrome, typically within 1 to 2 weeks of CAR-T cell administration, which coincides with the CAR-T cell expansion. And to date, several studies have already shown and reported that the CAR-T cell recipients who develop a severe CRS had also higher Cmax as well as a greater AUC. And here on the right bottom, you can see an example analysis plots, which was included in the ABECMA submission, which Satara supported, where a logistic regression model was applied to understand the relationship between the cytokine release syndrome rate and the exposure. And here for the exposure, the AUC was used. In addition to the CRS, we also need to evaluate the neurotoxicity, so the ICANS, as well as other adverse events which may occur, and they can be assessed in a very similar fashion as to the CRS using logistic regressions. To assess efficacy, we assess the relationship between the response rate and the exposure for different CAR-T cell doses via, for example, again, a logistic regression analysis, as shown here in the plot on the right, again, taken from the ABECMA submission which Atara supported. And in this analysis, it showed that with increasing AUC, we can actually achieve a greater response rate. And we can also look into time to event modeling to assess progression-free survival, and another analysis, for example, to predict the duration of the response. And what is interesting in these exposure response analysis is to identify the sweet spot. As you can see in that plot on the right top panel, at one point, the response rate flattens out. There's this Emax-shaped curve. So it doesn't mean that if you increase your exposure and your dose, your response will increase infinitely. At one point, you have maxed out your response, and it's really important to understand the sweet spot. The exposure parameters which are used for the exposure response analysis are usually obtained from a non-compartmental analysis or a POP-PK analysis on the cellular expansion data. So we derive the AUC as well as the Cmax or the Tmax. So the FDA has shown huge interest in the relationship between the safety and the efficacy markers to your dose response. So this is something we definitely want to look into before we submit the packages. If you go to the next slide, we also need to take into account some key factors that can affect the cellular kinetics. We have intrinsic factors as well as extrinsic factors which can affect your cellular kinetics. Intrinsic factors being patient demographics, the age, the gender, the body weight, or patient characteristics such as antigen load, tumor burden, or the CAR-T cell fitness. Also, as we just heard, cytokines and pharmacodynamic markers may affect the cellular kinetics, as well as immunogenicity markers. And for allogenic CAR-Ts, we need to think around the HLA matched versus a non-matched donor. Extrinsic factors, one factor is the dose itself. Is it body weight adjusted or is it a flat dose? And then other medications such as the lymphodepletion, the bridging therapy, the tocilizumab use, or the steroid use has been seen to affect the cellular kinetics. As well as that, we have certain factors in our product, such as the CD4-CD8 ratio, as well as the composition of the manufacturing parameters, the CAR construct, as well as the transduction efficiency, which can actually influence your cellular kinetics. And here, in this figure on the right, which was included in the summary basis of approval for CARVYKTI with the EMA, a forest plot was used to illustrate the assessed variability in the Cmax driven by various intrinsic and extrinsic factors as identified by using a population PK model analysis. So on the next slide, we are highlighting a few aims of CAR-T cell modeling. So what is the aim? First and foremost, CAR-T cell modeling can be applied to dose optimize our therapy to support safety and efficacy. Statistical models can be used to predict safety response, such as the CRS, but as well as to predict the long-term overall patient response in terms of survival based on the CAR-T cell dose. Cell modeling can also be used to improve the drug product, such as the improvement of the CAR construct. We can examine the CAR signaling kinetics and the T cell activation of CAR-T cells and compare these using modeling and simulation to the natural response of the TCR in the human immune system. And statistical models can also be utilized to link the drug product composition to the pharmacodynamic endpoints and the clinical outcome. We can also use CAR-T cell modeling to support questions around cost effectiveness, because these cure and survival models are used to predict the best dose at the patient population level to ultimately determine cost effectiveness, which can be sensitive to long-term remission rate and drug pricing. So our pharmacodynamic pharmacokinetic analysis of CAR-T cells can be utilized to optimize cell therapy product, the dose, and the lymphodepletion regimen to improve safety and efficacy. We can predict the expansion and persistence of CAR-T cell therapy. We can explore the interplay between the CAR-T cell dynamics and the tumor burden. We can predict the cytokine release syndrome severity and its biomarkers to facilitate the early patient intervention. We can explore the CAR-T-specific pharmacodynamic endpoints to link them then to clinical outcome to support submissions. We can identify a certain cell dose for a certain patient condition to achieve optimal therapeutic outcome. We can capture the quantitative impact of drug-specific parameters such as the CAR construct. And last but not least, we can use our established models to predict an optimal CAR-T cell dose in a special population, such as, for example, pediatrics. Next slide, please. So here are two pharmacokinetic models which describe the multiphasic kinetics of CAR-T cells, which an initial exponential expansion followed by B-exponential contraction and a gradual decrease of CAR-T cells that can actually persist in the body for months or years. So Stein and colleagues have actually developed a cellular kinetic model for Kymriah, which was the first nonlinear mixed effect semi-mechanistic model, which has captured the three distinct cellular kinetic phases: expansion, contraction, and persistence, as illustrated in this figure. This model also investigated the impact of tocilizumab and steroids on the rate of CAR-T cell expansion. This model was followed a few years later by a model developed by Ogasawa and colleagues, and Zatara was involved in this model to describe the cellular kinetics of Breyanzi. In this model, again, the cellular kinetic was described with three distinct phases. In this time, it was a lag time, a growth, and a biphasic decay phase. And the lag time was included to describe the cell expansion, or prior to the cell expansion, to describe actually the initial cell distribution and acclimatization of the CAR-T cells in the new environment. This model also investigated the impact of the tocilizumab and the steroids on the CAR-T cell expansion. On the next slide, we see a quantitative systems pharmacology model of CAR-T cell therapy, which was utilized to quantify the relationship among the CAR-T doses, the disease burden, and the pro-inflammatory cytokines. In this model, the infused CAR-T cells were activated after they interacted with the B cells. The activated CAR-T cells were also allowed to convert to memory cells, which periodically got converted back to short-life activated CAR-T cells to kill the B cells, which was used as a surrogate for the disease burden. In addition, the activated CAR-T cells also drove the secretion of the pro-inflammatory cytokines, the interleukin-6, 10, and the interferon gamma, into the blood to induce the inflammatory responses and the cytokine release syndrome. So this model has actually shown that the expansion of the CAR-T cells and the elimination of B cells are more highly correlated with disease burden than the administered CAR-T cell dose. So after successful treatment with our CAR-T cell therapy, we need to monitor the patient. Next slide, please. And before talking about the monitoring, we would like to introduce a poll question. So the question we have is, do you give a flat dose or do you adjust to body weight or body surface area? Okay, so the answers came in for the flat versus the body weight based dose. And we see that the majority is actually dosing adjusted to body weight or body surface area. And I think we have one more poll question to come before we move on. And that question is about the HLA matched subjects. So if you are in the allogeneic CAR-T cell therapy space, the question is if you do use HLA matched subjects or not. So we have two answers here, yes or no. Okay, here are the answers. So most of you who are using allogeneic CAR-T cell therapy are actually looking for the HLA matched subjects. We're going to talk a little bit about it in the next section when we talk about the monitoring. So monitoring on the next slide, we're going to start with the safety monitoring. So we have some key adverse events which are associated with cell therapies. So there is the cytokine release syndrome, which we mentioned a few times throughout the presentation, which is frequently observed within the first 7 to 14 days post-treatment. And this can be observed in an increase in the pro-inflammatory cytokines, including the interferon gamma, TNF-alpha, interleukin-6 and interleukin-2, as well as chemokines such as the MCP1. The symptoms are ranging from mild flu-like symptoms to severe life-threatening inflammatory responses, such as fever, confusion, and seizures. And tocilizumab, which is an anti-interleukin-6 therapy, is frequently given to manage the cytokine release syndrome, as it acts as a cytokine blocker. And as already shown during the modeling section, we can actually assess the potential impact of tocilizumab on the CAR-T cell kinetics using our pharmacometrics models. Another key adverse event is the neurotoxicity, these ICANS, and they present in headache, lethargy, tremor, and delirium. And the ICANS can occur in concurrent with the CRS, and they are generally also reversible with the use of tocilizumab. However, ICANS can also be presenting a little bit later, and then they are usually be managed by steroid use. And again, the use of steroids can be assessed on the cellular kinetics using our pharmacometrics models. Another key adverse events we need to keep in mind is the immunogenicity, which can be driven by the antigen recognition domain from the murine monoclonal antibodies that may be immunogenic in humans. And the immunogenicity can actually affect the CAR-T cell expansion and persistence and the overall clinical response, because the generation of the anti-CAR antibodies may lead to clearance of therapeutic CAR-T cells, which can increase the likelihood of the tumor relapse then, and then also lower the CAR-T cell efficacy upon reinfusion, which is becoming more and more of interest, especially with the development of the Allogeneic CAR-T therapies. So immunogenicity can potentially lead to resistance to repeat dosing. On the next slide, we touch on the monitoring of our efficacy. We monitor the efficacy by looking at typical endpoints such as the best overall response, partial responder, no responder. We can look at the progression-free survival as well as the duration of the response. And all this can be assessed using pharmacometrics models via logistic regression, time to event models, or Kaplan-Meier curves. Here are three examples which have been included in recent submissions. To the left bottom, we have the analysis which was included in the ABECMA submission supported by Satara, where we applied logistic regression modeling to understand the relationship between the response rate and the exposure. In the top right is an analysis which was actually performed by the FDA, included in the Tecartus submission, where the FDA looked into the duration of the response using Kaplan-Meier curves by exposure. And at the bottom right is an analysis which was included in the Yescarta submission, where box plots were used to identify responder rates versus exposure. Next slide, please. So what is the future? Next slide, please. So how can we improve the patient journey moving forward? Definitely by trying to reduce the complexity and the time to treat the eligible patients, by improving the safety profile, by starting to target solid tumors, as well as to think around pediatric therapies. Some potential solutions are already on its way. We're already looking into allogenic therapies, we're looking into the CAR natural killer cell therapies, into the T-cell receptor modified T-cell therapies, as well as that we start looking into the gating strategies, these or and not technologies. Next slide, please. To wrap this up before some Q&A on the next slide, we are highlighting some key takeaway points from this seminar. So getting the lymphodepletion regimen and the CAR-T cell dose right is crucial. Modeling and simulation can help to effectively support the development process and regulatory approval of cell therapies. And consider applying modeling and simulation early, ideally pre-IND, in your development programs. With this, I would like to thank everyone for your attention, and we are open for questions. Thank you.
Wonderful. Thank you so much, Miriam and Catherine, for that excellent presentation. As Miriam said, we are inviting our audience to put their questions in the questions panel for our speakers. And it looks like we have a couple already. So for Catherine, someone wants to know If autologous treatment isn't feasible, can a patient easily be switched to an allogenic treatment?
Okay, so that's a great question. I guess it depends on why the autologous treatment isn't feasible. If it's because the patient doesn't have sufficient T cells to enable leukapheresis, but otherwise that they are viable or eligible for a T cell therapy, then yes, that patient should be able to receive an allogeneic treatment. And that's one of the real advantages of allogeneic treatments, is that the T cells come from healthy donors, so they're much more robust and can be used in some of those more sick patients.
Thank you. Miriam, someone wants to know, can you tell us why the AUC 28 days and why AUC beyond 28 days is not expected to be associated with efficacy?
Well, so far, that's a really good question. Thank you, first of all, for that. So far, we have actually seen good correlation for the cell expansion. And as we already also saw in the systems model, the cell expansion was actually in the end linked to the clinical outcome and to the treatment of the disease burden. But I do think that more investigation, understanding PD markers, and correlation between early PD markers to clinical outcome are still needed and under investigation and are important.
Okay, thank you. Looks like we have another one for Catherine. I know it's a wide topic, but can you say something about the preclinical experiments that drug developers should do before they go into first in human?
Yeah, so I would say in general terms, the expectations for cell therapy are similar as for other drugs, in as much as you need to have an appropriate toxicology package, you need to understand the cellular kinetics, you need to know about the biodistribution, so where the cell therapy is likely to end up, and you need to demonstrate some confidence that your cell therapy is going to show some efficacy when you go to the clinic. What I would say for cell therapies is that it's really important when thinking about your first in human, it's really the safety aspects that are important. And we heard Miriam talking a little bit about CRS there, for example. So when it comes to selecting the first in human dose, for example, it's really the safety aspects that are really driving that dose selection for the first in human study.
Great. Thank you, Catherine. So question, LDC is well established in hematological malignancies, but it's been an area of debate in the solid tumor space, and a few developers have shown that low dose or no LDC is equally effective. Can you elaborate on that?
Is that one for Miriam?
Whoever would like to answer that.
Definitely, I think LDC are referring to lymphodepletion regimens,
Yeah.
right? So I take this. So lymphodepletion has shown in CAR-T cell therapy to be correlated to cell expansion. So higher lymphodepletion regimens have been linked to a favorable cytokine release syndrome, as well as to the favorable outcome in response rates versus the lower lymphodepletion has shown actually an unfavorable result. And hence, we do need to optimize the lymphodepletion regimens. And also now with the Project Optimus, the FDA is really looking into this and trying to understand the doses, the dose combinations, especially when we go to Allogeneic, and now including drugs like monoclonal antibodies, which were approved for a different indication and not for lymphodepletion. So there has been evidence shown that lymphodepletion is linked to cellular expansion. And as we saw in the systems model, the cellular expansion was in the end linked to the B cell killing and to the tumor burden rather than the cell dose.
Thank you, Miriam. So Catherine, we got a question for you. Can a patient reject their own modified T cells in an autologous treatment? What are the chances we can know beforehand that this could happen to a patient?
So what can happen in autologous treatments is you can get immunogenicity. So that's basically the development of ADAs, which is essentially where your cell expansion, you can see a drop in your cells because you're getting that immunogenicity effect. You can also get in patients with autologous treatments, you can get basically tired T cells, if you like, where you don't get continued expansion over time. So it's not really a rejection, I would say, but you can get some occurrences within autologous treatments that you wouldn't see with allogeneic treatments. You don't necessarily know that beforehand. There aren't any identified pre-specific factors where we know that may or may not happen, but we're early days, you know, it's evolving. Maybe we will start to pick up some of those factors as we learn more.
Absolutely. Thank you. Pose this question to whoever who'd like to answer it. Can CAR-T be used in all diseases eligible for cell therapies, or is it available for only a few diseases thus far? And then there's a second part. Does the dose optimization depend on the disease?
So I think it's still not for all diseases at this point, but I let also Catherine elaborate on that a little bit further. The dose optimization can definitely depend on the disease, and hence it is important that the dose is optimized, keeping in mind the underlying disease as well. And the beauty of these pharmacometrics models is if you then apply it to a different disease status, you can actually use the established models to predict the optimal dose for another patient population and another disease area, because we can include disease-specific parameters as covariates in our models. Catherine,
Great.
feel free to add anything if you.
Sounds good to me. Thanks, Maria.
Awesome. So I think, Miriam, you talked about lymphodepletion, so I'll pose this over to you. In case that there's a shortage of fludarabine, what would you say is next in line in terms of chemotherapy to fit with, is it cyclophosphamide?
I know that there is bendamustine, which has been used a couple of times also in case there is a shortage with the flu and the cyclophosphamide.
Okay, awesome. These are great questions from the audience. Miriam, you mentioned Project Optimus when you talked about lymphodepletion dosing. Does this also apply when lymphodepleting with fludarabine and cyclophosphamide?
No, it doesn't apply then, because that has already been established for lymphodepletion. It's only if you use a new molecular entity, such as, for example, you include the antibody.
Okay, great. Catherine, can you tell us how many cell therapy projects Certara has worked on, and were any of them submitted to regulatory agencies?
Oh, that's a great question. So let me think. So over the past, let's say 18 months, I think we must have worked on at least 15 to 20 different cell therapies. So not just CAR-Ts that we've talked about today, but also some of the other technologies that we touched on a little bit. So TCRs, NKs, some of the other new technologies such as the gated technologies. And yeah, in terms of submissions, so we support across all phases of development. So we've been involved in those early interact submissions. So that's the informal, non-binding CBER interactions that you have pre-IND for innovative therapies. Then also pre-IND, IND submissions. And then if the question was more focused on BLA submissions, then yes, we have, and the couple that we can mention because it's in the public domain is the two that Miriam talked about, which was Breyanzi and Beckma, that we helped in terms of the submission. So we really looked at the relationship between the cellular kinetics and the efficacy and the safety to understand what's really driving those to help support the dose selections.
Thanks, Catherine. It looks like we've got one final question from the audience. It's from Miriam. Miriam, can you discuss how you think Project Optimus and how it specifically relates to CAR-T therapies and what those developers should know?
Yeah, it comes back to the lymphodepletion regimen, right? So the FDA is really curious and interested in optimizing treatments, especially in oncology. And the lymphodepletion regimen is a key component. And the problem is that we are now starting to use in allogenic therapies treatments which have not been approved for lymphodepletion. So they don't have lymphodepletion in their regimen, so the approved dose is for a different indication. So hence, we need to look into the dose finding in order to get the approval, like the lymphodepletion, either into the label of that specific drug or into the CAR-T cell label. So hence, this falls into Project Optimus and will be reviewed by that team by the FDA.
Fantastic. Well, thank you so much, Miriam and Catherine, our presenters for today. And thank you to our audience for their attention, their time, their wonderful questions. On behalf of Certara, I'd like to thank you for attending this presentation. This concludes the webinar. Goodbye and have a great day.